My name is Rachel Gordon, and I am an assistant professor at the University of Pittsburgh.
And my name's Haley Cosgrove. I'm a postdoctoral associate at the University of Pittsburgh.
We are excited to share with you an overview of our recently published JCI Insight paper from our graduate work in Mark Schlumchek's lab.
NADPH oxidase in B cells and macrophages protects against murine SLE by regulation of TLR7.
Systemic lupus erythematosus is a multisystem autoimmune disease marked by loss of tolerance to self and rampant immune activation, resulting in damage to target organs, as shown on the diagram on the right. A hallmark feature of lupus is the loss of tolerance to nuclear antigens and the formation of autoantibodies to nucleic acids and nuclear protein complexes, as illustrated on the chart on the left.
The NADPH oxidase complex, otherwise known as NOX2, is a group of transmembrane and cytosolic enzymes responsible for the respiratory burst that is crucial to eliminate microbes. However, it's also been shown that loss of NOX2 function exacerbates SLE in murine models and is also associated with lupus in human patients. For example, chronic granulomatous disease, or CGD, results from loss of function mutations in key components of NOX2. Male patients with X-linked CGD are characterized by loss of function in CYBB, a gene that encodes for a critical component of the entire NOX2 complex. It's also been shown that loss of function polymorphisms in NCF1 and NCF2 are associated with SLE across multiple cohorts.
Until now, the cell lineages in which NOX2 regulates autoimmunity were unknown, as NOX2 has critical functions in neutrophils, macrophages, T cells, and B cells. Moreover, the mechanism underlying this protective effect has remained unclear. The goal of this study was to identify the cell lineages in which NOX2 deficiency drives lupus and the mechanisms by which NOX2 constrains autoimmunity. Let's dive into the data. Here, we used in vivo chimera and conditional knockout approaches to delete CYBB, the gene encoding a critical subunit of the NOX2 complex, in specific cell subsets of MRL, FAS, LPR, lupus-prone mice. We first generated conventional bone marrow chimeras using NOX2 deficient or wild-type donors and wild-type recipients. Hematopoietic NOX2 deficiency is sufficient to decrease survival in female MRL lupus-prone mice. The absence of NOX2 in the hematopoietic compartment worsens glomerulonephritis and interstitial nephritis. As NOX2 is highly expressed in macrophages, we wanted to understand whether NOX2 mediates its protective effects in lupus by its function in this cell type. We used a conditional knockout approach and a novel bone marrow chimera strategy where we deleted NOX2 in macrophages. We will show you data from the mixed bone marrow chimera cohorts. The details of this chimera strategy can be found in our paper. NOX2 deletion in macrophages exacerbates glomerulonephritis, as seen in the left-hand panel, and interstitial nephritis, as seen in the right-hand panel. However, NOX2 deficiency in macrophages does not increase the severity of glomerular and interstitial nephritis to the degree that we observed in the global knockouts that we have previously published. As the absence of NOX2 in the myeloid compartment did not fully mirror the severity of disease observed in the NOX2 global knockouts, we then explored the role of NOX2 in the B cell compartment using a conditional knockout approach. Surprisingly, the absence of NOX2 in B cells was sufficient to drive worse proteinuria, as seen in the left-hand panel, increased glomerulonephritis, as observed in the middle panel, and lastly, increased interstitial nephritis, as seen in the right-hand panel. We want to pause here to summarize data I have shown so far and additional data that we present in our paper to highlight in what cell types NOX2 regulates systemic autoimmunity. NOX2 deficiency in the neutrophil has no impact on lupus activity. NOX2 deletion in macrophages worsens lupus, but not to the degree that we observe in the global knockouts. NOX2 deficiency in the T cell compartment has no impact on disease. Surprisingly, the absence of NOX2 in the B cell drove rampant lupus nephritis, nearly capitulating what we see in the global knockouts.
After making these interesting discoveries, we next wanted to know what is the mechanism by which NOX2 protects from systemic autoimmunity. One clue comes from previous data from our lab showing that NOX2 knockout mice actually have increased anti-RNA and anti-Smith autoantibodies, and it is known that these autoantibodies are dependent on TLR7. So, we hypothesize that NOX2 may protect from systemic autoimmunity through the regulation of TLR7. We first wanted to test this hypothesis globally to determine if global deletion of TLR7 could rescue the exacerbated SLE seen in CYBB knockout mice. As you can see here, when TLR7 is deleted in CYBB-deficient MRL-LPR mice, there's improvement in parameters of kidney disease, including proteinuria, glomerulonephritis, and interstitial nephritis. Since in this paper, we have defined a role for NOX2 in the B cell by showing that B cell intrinsic NOX2 deficiency exacerbates SLE, we then wanted to test if the NOX2 TLR7 relationship is in fact also B cell intrinsic. Thus, we deleted TLR7 in B cells in globally CYBB deficient MRL-LPR mice to determine if B cell intrinsic TLR7 deficiency could rescue this exacerbated SLE. As you can see here, deleting TLR7 specifically in B cells on a CYBB deficient background does in fact improve parameters of kidney disease, including proteinuria, glomerulonephritis, and interstitial nephritis. After testing this genetically, we then wanted to dive deeper into the mechanism. We hypothesized that if NOX2 is regulating TLR7 in the B cell, it could be via a negative feedback loop whereby NOX2 ROS dampens TLR7 signaling. Because NF-kappa B is activated downstream of TLR7 stimulation, we tested our hypothesis by quantifying NF-kappa B nuclear translocation in NOX2 sufficient versus deficient B cells downstream of TLR7 stimulation with or without BCR activation. We show here that CYBB knockout B cells do in fact exhibit greater NF-kappa B translocation after TLR7 stimulation. Importantly, as we further show in the paper, this is specific to TLR7 but not TLR9 stimulation in B cells.
In the second half of this talk, we show that TLR7 deletion in B cells ameliorates nephritis in NOX2 deficient lupus-prone mice.
We also show that NOX2 knockout B cells have increased NF-kappa B pathway activation after TLR7 but not after TLR9 stimulation.
NOX2's selective regulation of TLR7 signaling is another piece in the puzzle that helps explain the well-established finding that TLR7 drives lupus, but TLR9 is protective in this disease.
Thank you all for your attention.
A special thanks to our co-authors for their help and support. We hope that you enjoy the paper.